Okay, so while we're getting set up for the next speaker, I'm going to introduce him. Our next speaker is Kevin McDonnell. He has a PhD in computer science from the University of Galway in Ireland, and he is now working at PTI as a computational scientist, and he's going to tell you what he has been doing the last 2 years.
Cool. Thank you, Corinna. So yeah, I'm going to talk about some of the technologies we've been developing at Parallel Square Technology Institute, and just a brief overview of what that is. I was asked a lot yesterday, and you're going to see some of the people floating around. So we're a non-profit focused research organization. And one of the goals of PTI is to develop technologies that can increase throughput for proteomics. And so I'm going to talk about a couple of those today. And just off the bat, I want to acknowledge one of those is a project led by Harrison Specht in terms of new mass tags, and then also another project led by Jason Dirks in alternative domains of multiplexing. So to start off, we'll describe throughput. We want to increase it. Well, what is it? So we define it as the number of parallel peptides times the number of parallel samples in which we can identify those peptides. And if we want to maximize, say, the number of samples which we can analyze in parallel, typically we would do something like maybe a data-dependent acquisition approach. And in this approach, we take an MS1 survey scan. In that scan, we will send particular precursors for fragmentation, and we can use those fragmentation spectra to identify them. We can combine this with something like TMT, which can get 35-plex. So you can, if you identify a peptide, you can quantify it in 35 samples simultaneously. However, if we want to parallelize the number of peptides or maximize the number of parallel peptides we acquire, we might do something like data-independent acquisition. In this, we again take a survey scan, but instead of selecting particular peptides, we actually send these set windows. And so multiple peptides are sent for fragmentation. And so this is great because we can actually, for a single scan, we can identify multiple peptides. However, that creates more complex spectra. But current tools are well able to deal with this kind of complexity, but that can change as we actually multiplex even more. So as described by Dirks et al. In 2022, with 3-plex is kind of some of the state of the art. I know Proteome Sciences have some more tags out, and there are other technologies like diethylation, but we're working towards developing N-plex DA. Can we maintain the level of parallel peptides and yet scale to the number of parallel samples we can get with maybe a DIA, or excuse me, a DDA approach? And so just to talk a bit more about what is plex DIA, so this is where we multiplex in the mass domain. So for a particular peptide, we attach a molecule, so something like this ImTrack molecule. And so when we identify a peptide, we will have a channel that has the molecule attached, but in the next peptide, we'll have a different isotopes incorporated into the molecule, so we get this mass shift. And so we can identify 3 samples here together with just mass offsets between them. And what has been historically done is separating or inducing these 4 Dalton mass shifts, and this to account for the isotopic envelope of the peptide, so to minimize the overlap. And so for this particular example, there are 2 limitations. One, it's currently only a 3-plex if you're using ImTrack. And so we need maybe bigger molecules that we can incorporate more isotopes, get greater plexing. But if we could reduce this 4 Dalton offset, then we could actually also increase the multiplexing for the same molecule size. And so that's where we're going to go. So in this talk, like I said, we're going to talk a bit about some new tags we've been developing, a project led by Harrison Specht. I'm going to talk about a new dimension. So I've just mentioned multiplexing the mass domain. What if we multiplex in the time domain? And I'm going to talk about JMod, which is a piece of software we've been developing at PTI, which enables searching and analyzing the data that we acquire with these, because the way this data is generated is not currently searchable by the current algorithms, so we created our own. So to talk a bit about the story of developing new tags, so there are a couple of criteria we're looking for. So we set up the split pool synthesis approach where we can screen lots of different molecules, and with the goal of maintaining the number of parallel peptides. So we don't want to attach a molecule to our peptide and make it harder to identify. We want to maintain the quality of the spectra we produce, make it still easy to identify, or perhaps improve that. And we also want to allow the molecule to parallelize the number of samples, like I said. And so step one was screen all these different tags. So each point here is a different tag attached to both this peptide and this peptide. And these look at the scores, the Andromeda score in this case, for all of the different molecules when they're of the two peptides when the different molecules are attached. And PSM tag here is the molecule that we settled on, as it showed high scoring for both of these peptides. But to give, I guess, a more comprehensive evaluation, well, you can compare it here to label-free. So just to step through this graph really quickly, each point here is a different peptide, and it's on the x-axis, we're showing the ratio of precursor intensity. So how abundant was the peptide when it was labeled with the tag versus label-free. So if a point is here on the right-hand side of this graph, it means it was more abundant when the tag was attached, or it was observed a greater intensity. Then we have the same ratio, but with score. So on the y-axis, so if a point is above this line, it means when the tag was attached, it scored better than the label-free counterpart. And you can see that most points are scoring better, and most points are actually more intense. So this tag is exhibiting these positive spectral qualities when attached to a molecule compared to a label-free scenario. And just to explain maybe what that would mean. So here we have a label-free peptide. And so when we want to identify it, we send it for fragmentation. It generates this series of ions. So the C-terminus ions are the Y ions. And you can see for this particular case, we have the full series of Y ions were observed. And we also have some B ions were observed. And this results in a hyperscore of 34. And without going into detail of a hyperscore, it just encapsulates the intensity that was observed and the number of fragments. More intensity, more fragments, better score. Here's the same peptide, but with PSM tag attached. And the first thing to note here is we're getting a higher score. And we can look into why that is. We can see, again, the full series of Y ions is observed. But now we're observing more B ions. And so you can see how the Y ions are observed in the same positions, but the B ions are shifted as this is an N-terminal tag in this case. And so for this particular case, it might be super important because we already have the full series of Y ions, but this is particularly powerful for technology such as de novo sequencing or maybe PTM localization, where you want as many fragments from as many of the fragmentation sites as possible. And you can read the preprint if you want to see more on that analysis. But just to explain this more completely, here we have B ions on top and Y ions at the bottom. Our blue bars indicate the frequency with which we observe these particular B ions in label-free spectra, and in orange with PSM tag attached. And you can see how PSM tag is helping the observation of these B ions systematically in our spectra. And this is the particular molecule itself. And so, like we said, this is great. We're exhibiting positive peptide properties. We're not going to sacrifice the number of peptides we see by attaching this molecule. We may, in fact, enhance the number through these positive properties it's exhibiting. But each star here indicates where we can introduce a different isotope. And this allows us to multiplex in the mass domain with this particular tag. If we maintain this limitation of 4 Dalton spacing, we can get to a 5-plex with this particular molecule. But if we were to relax that constraint and just require 2 Dalton spacing between the different channels, we can get to a 9-plex. But currently, there aren't software available that can search this type of data. And just to talk about the effect of this, so we're talked about this in a DIA. We're parallelizing the number of peptides in a particular spectrum. We may have different peptides in our library. All of these are present. And so this is a cartoon, but it illustrates the case. Some peaks will be distinct, will be only attributable to one particular peptide. Some peaks will actually be a superposition of the contributions from multiple peptides if they share that fragment or very similar M/Z properties. And so just to look at how much this happens in a label-free case, if we look at, say, here for a 16 Thomson window size, which is not that big, you can see that most peptides have fewer than 70% of their peptides are distinct. And so even in the label-free case, we're seeing this overlap in terms of fragments. But we want to push towards greater and greater multiplexing, so we're putting more and more peptides into these spectra. And so this is only considering monoisotopic peaks, it's not considering isotopic overlap. And you can see how we're driving the number of distinct fragments per peptide down towards 0. So this is including the 5-plex and the 9-plex. And so as we move towards greater and greater multiplexing, we need to think about how can we handle the type of data where this overlap is increasing further. And so you can think of different ways. Some current technologies throw out overlapping signal. I think it was talked about yesterday. There are others that assign it to a single precursor through competition. And I guess both of these methods, as we drive more and more towards this greater multiplexing, you're maybe getting rid of some of the signal you want to use. And so there's an alternative maybe where we can model this overlapping signal. So just, okay, make the assumption it comes from multiple sources. Can we try and deconvolve those sources into the individual contributions? And so this is the approach we took. And so in doing so, we developed a piece of software called JMod. It is open source, it's available on GitHub, and there's a preprint as of yesterday online. So I encourage you all to check that out, as there is for all the different technologies I'm talking about today. And with the goal of supporting multiplexing, thereby enabling this increased throughput. And it's, as I said, built on this assumption that we can model this spectrum as a superposition of the different library precursors. This is not a new approach. This was done by Spectre. It's done by Chimeras. But it's something that needs to serve as the baseline for a model as we move towards greater multiplexing. So that was a cartoon example. Here's a real example of JMod. So these are just monoisotopic peaks, so they're just filtered for clarity, and they contain 2 very similar peptides. And you can see again in a real example where distinct peaks can be attributed to the individual peptides, but it can account for this overlapping signal and attribute that signal to the different peptides it came from. But what current algorithms can't do is model this overlap induced by plex DA. If we were to when we start having overlapping fragments that are induced by these shorter offsets in the mass domain, we don't currently have technologies that can deal with that. And so I mentioned this 4-Dalton spacing, and it's like, why do we care about this? So just to briefly labor on the point, the reason we have these is because we don't want, or historically we wanted to minimize the isotopic overlap between the channels. And just to give a brief explanation, it's very clear probably to most people, carbon-13 occurs naturally. And so for a typical maybe peptide, you get a distribution like this. So the monoisotopic peak has no carbon-13. These are where we have 1 carbon-13 in the molecule, 2, et cetera, et cetera. This follows a binomial distribution dependent on the number of carbons. And here you can see there's 4 distinct peaks, hence we have 4 Dalton offsets. For larger peptides, which are maybe observed less frequently, but you can see how 4 Daltons is not actually enough for these kind of peptides. But like I mentioned before, PSM tag allows this 5-plex if we maintain this 4 Dalton spacing. But what if we could halve the number of spacing we require, so down to 2 Dalton, we could effectively double the throughput with this same tag. And so that's what we did. We evaluated JMod with its ability to model this overlap induced by these overlapping isotopes. And we also just compared it to label-free and to a 1-plex to give you an indication of how this the performance is doing. So to visualize this, here is a real observed MS1 signal from this very nice long peptide. And you can see the different isotopes are distinct here, and they're stacked like a train back to back. And so when we use JMod, we can deconvolve this signal into the individual contributions. And you can see how here, because it's a long peptide, we have signal coming from one channel into the next. But this can be, with the joint modeling approach, or this, we can deconvolve this signal into the individual contributions. If we relax the constraint to 2 Dalton offsets, now we get this very non-distinct isotopic overlap. It looks something like you might see a protein isotopic overlap. But applying this joint modeling approach, you can see it's made up of all the different channels. And this is real data fitted MS1 coefficients that come from JMod for this peptide. And so we can look at the identification. So this is 200 picograms per channel, so approximating single cells, labeled with PSM tag or labeled free and acquired on Orbitrap Astral. And just to step through this graph, you can see the number of proteins here identified in label free context with just a single PSM tag channel. And then as we increase the multiplex, we can see we're scaling the number of precursors. Our protein data points we are acquiring in a single run. And so maintaining this depth, or almost maintaining this depth of coverage as we increase the plex size. And we can compare this to Diane. So Diane is the leading search algorithm in the plex DIA space. And you can see we're performing comparably well for the label-free delta zero on the 5-plex, but currently JMod is the only algorithm that can deal with these overlapping isotopic envelopes. And so it uniquely enables this 9-plex setup. And as Alexei said yesterday, IDs are not everything. It's not about getting the most identifications. We want to care about the quality of the data. How good is quantitation? And so we set up this mixed species experiment, as other people have talked about, where we have constant inputs in different mixtures of human, and then we have variable amounts of yeast spiked in. And we evaluated this in the 5-plex and the 9-plex conditions. And we just label different mixtures for every different configuration of PSM tag, combine them, and then ideally the algorithm is able to give us back the quantities, particularly of yeast, the different relative abundances for the different mixtures. And so to visualize this first, I showed you previously this human peptide. So all of the input amounts are the same intensity. But if we have a yeast precursor, you can see how this changes. And so you can see here now there's some significant contributions coming from previous channels into lowly abundant channels if they're followed by lowly abundant channels. And so to start, we'll just evaluate in this 5-plex example. This is still separated by 4 Daltons. You can see JMod has given us very accurate quantitation here. So this is a 4-fold change delta zero. It's 4 times higher than delta 12. And we're able to retrieve this 4-fold change back pretty well. And then to maybe give a more systematic evaluation of the quantitation accuracy, we're going to collapse all the different comparisons into their mixture ratios. And so look here, A over B instead of channel over channel. And we can look at all the different all the different mixture ratios. And so we can see we can get accurate quantitation for 2-fold, 3-fold, 4-fold change. And this is as high as we've evaluated currently with this particular setup. Again, we can compare to Diane, which is the state-of-the-art in plex DIA. And you can see here we're actually providing more accurate quantitation for these mixture ratios labeled with PSM tags. So this is 5-plex data, so it can be searched by current methods. So next, obviously, we want to evaluate the quantitative accuracy of 9-plex data. Again, I showed you this human precursor previously. And it's somewhat easy or easier because we have a significant signal in each of the channels. But what happens here is it gets a lot more complex when we have these 4-fold or large fold changes between the different channels. And you can see here back to delta 12, again, if you look at the monoisotopic peak, it contributes less than half the signal to the monoisotopic peak that it's attributed to here. And so the question is, can we accurately quantify peptides with this level of overlap? And so these are the results from the 9-plex, 2 Dalton offset spaced for the different mixture ratios. And you can see we're still getting accurate quantitation for this data despite the overlap induced. And we can compare this to the 5-plex methods for the two, for both JMod and Diane. And you can see it's pretty comparable, pretty on par with the other 2 methods. So we're not really sacrificing quantitative accuracy. We're still maintaining those accurate predictions of the relative abundances of these yeast peptides in the 9-plex configuration. And we can focus in a bit more on, say, these 4-folds. So in this case, the largest fold change considered. If we look at those in the 5-plex and the 9-plex configurations, you can see for both here, we're getting, this is the delta 0 and delta 12, delta 0 and delta 12. We're getting a really accurate quantitation for both. It is to note, we are a slightly wider spread for the 9-plex data, but this is still very accurate quantitation with a tight distribution around the expected amount. And this is despite, as I said, this is quantitation coming from this delta 12 channel here, which for the monoisotopic peak is very little signal actually being attributed versus the delta 0 in this case. And so we're still able to maintain that quantitative accuracy. And now I'm going to talk a bit about alternative multiplexing domains. So I've described a lot of multiplexing in the mass domain and how PSN tag enables us to increase multiplexing in the mass domain. But a project led by Jason Dirks at PTI was exploring alternative multiplexing dimensions. And so analogous to multiplexing in the mass domain, we also acquire data in the time domain. And so we can multiplex in the time domain by inducing offsets in retention time to our different samples. And so how that works is, as our peptides typically elute off the column, we have a very high expectation of when they will elute, and the order particularly in when they will elute. And this can be improved with transfer learning, machine learning. And so we can get very accurate predictions of when our peptides will elute off the column. And so taking advantage of this, Jason stacked a second column spraying at the mass spectrometer, but with an induced retention time offset. So this one now is the same mixture or same peptides, but they elute consistently 4 minutes later with the exact same pattern. And this can be expanded to M columns. And so you're effectively multiplexing in the time domain by however many columns you spray at your mass spectrometer. And what is really, I think, the particular strength of this approach is that while it's similar to PlexDAA, it's completely orthogonal. So what you can do is you can combine the two approaches and you can get multiplicative increases in your throughput. So here you have a 3-plex, say, in your mass domain, and a 3-plex in your time domain gives you a 9-plex overall. And so you can get these multiplicative increases. And of course, it begs the question, well, I've just talked about 9-plex. I've talked about a time in the mass domain. I've talked about time-plex. Can we combine those approaches? And that would effectively give us a 27-plex. And so this is data acquired 1 nanogram per channel on Explorers 480. And you can see how we have label-free, then we're protein data points, 9-plex DA, and then you can see this 27-fold increase in the throughput for a single run. Here is just showing some of the chromatograms acquired by time-plex. So you have the human traces where you have this consistent quantity because they're spiked in at the same amount, and you have the variable quantities of the yeast. And so just to conclude, we've been developing technologies. So time-plex, it gives us parallelize the sample so too does PSN tag, but by combining these approaches, we can effectively get this multiplicative scaling of throughput, which is very powerful. And so to finish, just to acknowledge all the people that made this happen: Smith Futures, Converge Research, Griffith Catalyst, partners in Creagen and the Pentalutia Group, and of course, everyone at PTI, and particularly Harrison Spector, as I mentioned, and Jason Dirks, who have led those 2 projects that I talked about, and the whole team, it's been a complete team effort. So thank you all. And just to acknowledge, so as of yesterday, as I said, the 3 of these projects are now available on bioRxiv. They're available on GitHub. They're open source. So please check them out. These landing pages will give you all the information to the data, GitHub, and the preprints. And thank you very much for listening.
We have time for a few questions. Oh, okay.
Thank you, Catherine. Great talk and really impressive research. My question would be about if you've used MS1 and MS2 quantification, if you've also looked into MS2-based modeling, and if you have any expectations about how the more complex signal can actually help identification of these very lowly abundant peptides once we know that the signal doesn't come from 1 peptide copy, but actually from 27 peptide copies.
How that helps identification?
For example, yes. Kind of like stacking in TMT.
Yeah, I guess, and that's something we've observed. Now, there were other confounding factors, but arginine, typically the Y ions stack on top of each other. So for low inputs, identification becomes better for those kind of data points. So it's really powerful in that side of things. In terms of quantitation, we focus mainly on MS1 quantitation because we've been dealing with single cells. So we are currently working on improving our MS2 quantitation. but it's been mainly MS1 focused. But yeah, I guess that's one of the advantages of multiplexing is you can kind of almost, yeah, I guess boost your signal. But then alternatively, you need to have strong channel FDR to account for that. So you're not giving false identifications either.
Thank you.
So you mentioned that this PSM tags will help to boost the B ions. So, but I mean, though you're quantifying the protein at the MS1 level, but considering for even for identification, so if we see the spectral knot or the ions, so they never take the whole series of ions to identify the proteins. It takes maybe top 6 or top 5.
Mm-hmm.
So what is the advantage having a more of B ions with these tags?
The advantage in the context of DIA, what I showed you is a really nice peptide that had a lot of Y ions. That is typically not the case, particularly as we get to maybe longer ones. And Alexi talked to us about requiring at least 5 fragments. Well, maybe that's currently throwing out some signal, but if you can boost the number of fragments you're actually observable of your peptide, you are increasing the number of peptides you can now match confidently. What's really powerful about this kind of approach, though, is like I mentioned in terms of de novo sequencing, where again, that peptide we saw is maybe atypical in that it had the full series. De novo sequencing is very strongly reliant on having, the full series of ions, which is of fragmentation sites having a particular fragment present. And that is shown in the preprint that this tag is helping boost the full fragmentation site coverage in DDA peptides for de novo sequencing.
So this is timescale multi-column approach is also possible on the timsTOF?
On the timsTOF, yeah. So this is kind of, I guess, the next step. Some of the third-party software we're using to work with timsTOF data, it just didn't work out in time for this particular preprint, but it's not currently available for timsTOF data, but will be shortly. That's our main priority now, once this is.
How does the joint modeling take into account decoy peptides? Are those also modeled within the partial contributions?
Yeah, so the spectrum fitting fits both the targets and the decoys simultaneously. Yeah, I guess this is there's a Nathan who works with us has talked about, and I think it was done maybe in the Spectre paper too, how you could do an identification run where you just fit both targets and decoys together, and then for MS2 quantitation, remove the decoys and just fit those high-scoring targets to get better quantitation. But for the identification, yeah, we just fit them all simultaneously. And so, yeah, if decoys are attributed signal, they should be at the same rate as your false positives, ideally.
Just curious if the limit ahead is chemical or computational. How many maximum multiplex tags you can?
Computationally, I'm not sure if the limit is. Maybe in terms of how many ions you can get into the mass spectrometer might be more of an issue, but we haven't run into that. Another limit is we want to explore more is, I guess, the fold changes we can look at with this particular model. I showed a 4-fold change. We can still accurately model 4-fold change. What happens if you get to 100-fold change? Now the level of error maybe in your fitting, if you don't have the correct model of your peptide, does that now error proportional to actually the fold change you're observing? So these are things we still want to explore, but yeah.
Hi, great talk. Just quickly on the, a little bit more about how the joint modeling works. Are you fitting more of like a deterministic linear model on the isotopes that come out, or is there any probabilistic kind of noise you might be assuming, or is it kind of, would that be kind of under constraint, or maybe you don't?
Not currently, no. It's, yeah, I guess, a deterministic in that sense. And I guess, like I was saying to Brianna, having the correct model is super important, because one of the things we noticed was if you don't take into account the actual isotope distribution of the tag, that's a pretty significant contribution. So you have the incorrect model, you're going to get incorrect quantitation. So it's super dependent on having, the correct predictions of, say, your fragments of your MS1 data. And so with machine learning and approaches like Georg's alpha DIA or alpha PEPT, deep, you can get accurate predictions. And so that kind of, I guess, yeah, kind of, I guess, enables this kind of approach.
Yeah, and I wonder, like, if you were to take examples where, say, the fit didn't match your expectations, if you think there's anything to kind of to learn there from other things that might be happening, or essentially, I don't know, something that maybe you thought or is playing a factor that you didn't consider that you could learn from kind of cases where you have a lot of ions, but for some reason the model doesn't quite fit the spectra or something like that.
I guess we've looked into it somewhat. I can give you one example is if there's a peptide there or some species there that's not in your library, that's going to mess up your fit because that's going to be signal that you're attributing to somewhere else. So it's super important having the correct library for these approaches. But yeah, maybe there are some other interesting things we can see for sure.
Very nice talk, maybe not related to the software specifically. It's nice that it's open source though, I like that. But how easy is it to implement PlexDia? I mean, if I would like to do it in my mass spec lab.
Oh, I'm purely computational. You have to refer your questions to the mass tags team. They're scattered around here. They have worked a lot, I guess, on optimizing and making it simple. I think that's one of the things we want to do, is to make this accessible to everyone. So I guess I can't give you more detailed answer than this is something we're strongly looking at and trying to optimize and release protocols so that it makes it easier to do.
Great, though. Also, not a computational question, but if you run 2 or 3 or 5 columns, how significant is the hit on the sensitivity and the quantitation accuracy that you're observing in your data analysis? So you're using one mass spectrometer, you're using a trap, they're competing ions and so on. What do you see?
The effect of the heat, sorry? Or
the effect of multiple columns.
Oh, okay,
You're
excuse
making
me.
artificially increasing the complexity of the sample.
Yeah, yeah.
So-
One of the things I guess Jason has done great work on is if we are, I guess, acquiring multiple samples from the same column over time, you have to do this normalization, I guess, for all the different samples. But what happens when you have 3 columns spraying together, you have spray issues, variable spray. You need to normalize, I guess, the intensity that's been sprayed at the column. So that is just fitting like a lowest fit to actually, you can see the intensity will kind of fluctuate somewhat across the run, but having a fit, you can actually counteract that and normalize that. And in terms of then, I guess, globally quantitative accuracy, it is a bit lower currently for this data, but I was also comparing Astral versus Explorers, so maybe there was some of that involved, but it's things we're ironing out because I think computationally we can also make some improvements there.
And it's a nice concept, but how practical is that? Can you run hundreds and thousands of samples on 3 columns and for days and days?
Yeah.
I just want to make a quick comment here. There are 2 different things. One is the concept of multiplexing in time, and the other is particular implementation. Jason has done 2 different implementations for that concept. We think none of those is optimal. I think each one of them is sufficient to demonstrate the potential and to make us very excited about it. But we certainly envision possibilities of having multiple separations on a single column using isocratic separation, other approaches that we've discussed with you and Jared and other colleagues. I think there's a lot of space for innovation to make this a really robust product. At the moment, I would not recommend everybody go run TimePlex. I think that's a good thing for experts to do and get their hands on, and innovators in the technology to push it forward. But we foresee that the ultimate implementation of TimePlex is going to be different from the specific proof of principle demonstration that we've done. And we can also reduce flow rates to minimize the impact on sensitivity. And generally, our modeling approach is very much focused on dealing with the increased density of data points, modeling that so that we can minimize the impact of quantitative accuracy. And thanks to our friends who develop more sensitive, higher resolving instruments with higher capacity, we see a lot of space to keep pushing in that direction.
Nice concept. Thank you.
Okay, I think there were a few questions online. Can we see them here?
There were a lot of questions about commercial availability of the tags. I saw there were at least 3 or 4 asking that
Yes.
question. So I can chime in on this. We certainly would like to make these tags available to the community as soon as possible. And at the moment, you can immediately take our open source synthesis protocol, go to a CRO and synthesize it. We are also discussing with partners interested in licensing those tags and making them commercially available. And we would love to speed track this as much as possible. So that's the plan. Stay tuned.
Okay. Any? Oh, yeah.
So thank you. I have a technical question. Because there's a difference due to mass defect between the isotopes of carbon and nitrogen, as we know from TMT. Is it possible with high enough resolution to split the MS1 peaks into doublets and then doubling the multiplexing this way?
This is something we're looking into, exactly that. So actually taking advantage of that mass defect. And so, yeah, having them, I guess, offset by millidaltons as well as overlapping. And so you can, theoretically, if you introduce, say, enough nitrogens into your molecule, you can do this.
Okay. Any more? Yeah.
One of the biggest trade-offs that you have when you are labeling with TMT is a loss of dynamic range that is getting the larger, the more plexes you're using. So how big is this loss of dynamic range when you're doing plex DIA?
As I said, yeah, this is the next step to explore. So we looked at a 4-fold change. We didn't see a lot, but I guess, do you mean dynamic range in terms of different molecules or in terms of the same peptide analyzed? Same peptide. Yeah, I guess this is, yeah, like I said, if we have a 100-fold change, is the error if you don't have the perfect model of your data or there's some like noise in your data, that fold change now if they're overlapping is relative to the same size as maybe the error in your measurement. And so this is something we need to explore, but yeah, we don't have an answer for that, I guess, yet.
The one related thing that we can certainly share at this point is that there is no ratio compression as in the case of TMT. In the case of TMT, for any fold change, even if that is 50%, 2-fold, there is ratio compression, while in this case, we are quantifying ions that are specific to the sample and to the peptide and to the protein. So there is no systematic shift in the ratio. Quantitative accuracy is as good as for label-free analysis.
Any last questions before we close the session? No? Okay, then let's thank Kevin again.
Thank you.